going to try it, see what happens. Do people see a cover slide here?
It looks good. Yep.
Okay, thank you very much. I'm delighted to be here. I should just warn you, I've already had unstable internet flash across my screen. Even though I live in downtown Kingston, I'm not sure why that happens. I'm not living in a cottage, but I have 2 lines coming in, and I switched, I think, to a more secure line. If I disappear, give me a couple of minutes. I'll find a way to come back in, which has happened in lectures before. I realized when I saw the agenda, I slightly changed the title because I wrote that title in a hurry. It's essentially the same talk, slightly different. I added the longer summer is what I'd like to talk about, in that we are dealing in an environment where people say the winter is getting shorter. I look at it as the summer is getting longer. And that is actually new challenges, I believe, for lake management. And before I begin, I'd like to acknowledge I'm sort of the speaker here, John Smol, but these are colleagues of mine. Kaplish Balsamarian, we call him Cap, is the graduate student, the master's student who is actually doing much of the work on the Rideau Lakes as part of his graduate degree with me. Kathleen Ruland is a research scientist in our group, and she's involved in helping Cap and working on this. And Andrew Patterson is actually in the Ontario Ministry of Environment, Parks and Conservation, also an adjunct professor in my lab. He's at the Dorset facility up in Dorset, Ontario. But all these are contributors to what I'm going to be talking about today. Now, you're a lake group. I don't have to remind you that we need water. We refer to it as freshwater services. And everything we think perhaps first drinking water and agriculture and angling and hydropower. But of course, you're also in an area where a lot of these lower things happen, where you're using also lakes for recreation. And for that, you want to have high quality water. You don't want algal blooms. You don't want dead fish. And I just keep in mind, I always say this to my students, we often start talking about this, but it's a very human centric view. We also have to keep in mind, these are the real users of water. Things like the fish, the various types of invertebrates, plants, and so forth. So we keep that in mind. Having said all this, that how important water is, and it is important for us, but also the other organisms we share with the planet, we've not been very good at taking care of our environment, let alone our water. Amongst other things, we've been using the atmosphere as an open sewer. One thing that comes to mind is greenhouse gases, for example, but in addition, all sorts of other pollutants. And what goes up does come down eventually, things like mercury and metals and so on and so forth. And of course, a lot of that is affecting lakes. But we also do a whole lot of other things to our catchments, different types of mining, different types of point sources, types of overfishing, and so forth. So we really are dealing in what we refer to as a multiple stressor world. And to do that, so I'm an environmental scientist. I'm a limnologist. I work on lakes. I'm sort of the freshwater side of oceanography. And one of the biggest challenges we have in environmental and ecological work is the lack of long-term monitoring data. We live in a world of the present in many ways, but if you think about it, if you have dead fish or you have algal blooms, and if you treat it like a human disease, if you think of going to your family doctor, well, the first thing your family doctor is going to ask you is your medical history. He or she is going to want to know, what were you feeling like before you were sick? So your background, your baseline conditions, your natural conditions, if you like, your reference conditions. They want to know what were the symptoms as you started feeling sick. These are all sorts of important questions. They're trying to establish what is the disease, how bad is it, or are you in fact sick? The same thing as for example, a detective coming onto a murder scene. The first thing the detective wants to do is to try and reconstruct the history of how we got there. Well, if you think of a problem in a lake like a murder scene, that's more or less what we do. We try and reconstruct the environmental history to find out, how did we get to the situation? What went wrong? Why do we have a problem? And when it comes to environmental or ecological questions, these are the typical questions we're asking. We want to know, what were pre-disturbance conditions? We want to know, what was it like before European settlers came, for example? What was Lake Opinacon or Indian Lake like before? What was it like 200, 300, 400 years ago, before we added all these anthropogenic stressors? Another good example is acid rain. I cut my teeth when I was a young professor. The early work on acid rain using the approaches I'm showing here. Acid rain is a good example of how you need to know pre-disturbance conditions. Now climate change is the headlines. Back in the late 1980s, early 1990s, acid rain was the headline. The biggest question then was, have lakes acidified? Certainly, people like me said, of course they acidified. There were people in certain industries who were potentially causing the acidification, didn't like that answer. And the argument was made, the lakes are acid now. You couldn't argue the lakes were acid now. You take the acidity readings now. The argument was made, they were always acid. They're naturally acid. How do you show they're not naturally acid? The only way you can show they're not naturally acid is to go and find out what the acidity readings were like in the early 1800s before acid rain. Well, that's impossible. For one thing, pH didn't even exist, and pH was only invented in 1909 as a function, and no one was measuring water chemistry back in the early 1800s. So you had to show that the lakes indeed acidify. And the techniques I'm showing you here are the same ones we used then. You want to know, what is the range of natural variability? A good example here could be climate change. I don't think anyone can deny something very strange has happened to the climate, but the argument is made, oh yeah, but that happens all the time as you go in history. Well, we can show that that's definitely not the case. Have conditions changed? How much? How fast? When? Why? Each of these questions has a time or a temporal component associated with it. And that is one of the biggest challenges we have in environmental work. We don't have the monitoring data. In a recent paper I published late in 2019, I asked the question, how long are most monitoring programs? And I went to the International Journal Environmental Monitoring and Assessment. I looked at the year before, about 423 papers that dealt with aquatic systems. And I asked, how long were most of the monitoring programs in this journal in environmental monitoring assessment? Turns out that about 2/3 are less than 1 year in duration, and about over 90% are less than 3 years in duration. Now, that's not to say 3 years or less is not enough time to answer important questions. Of course it is. But 3 years or less of data is not enough time to answer those questions I posed on the previous slide. So what do? We have to find other ways of reconstructing the past. And the way we do is paleolimnology. It's a mouthful, but limnology is the study of lakes and rivers. Paleolimnology is the study of history of lakes and rivers. And the process is very straightforward. The bottom mud, the bottom sediments on a lake are a time machine. The surface is 2021. The bottom is when the lake formed. And in places like the Rideau Lakes, they're about 12,000 years old. It's when the last ice age was here. When the last ice age slowly left about 12,000 years ago, one of the legacies of that ice age was the holes left behind, which filled with water, and they're now the Rideau Lake system. So when the ice age left, that was day one. That was the time zero. And since that time, in most lakes in the Rideau Lakes region, there's now about 3 or 4 or sometimes 5 or 6 meters of sediment. It's a history book going back 12,000 years. The deeper you go, the older it is. Right at the base is about 12,000 years ago. Right at the top is today. And in between is a history of how that lake has changed. And those sediments come from a variety of sources. They come from outside the lake, things like pollen grains, contaminants, soil particles, all that's making its way into the sediments. Now, I'm a lake biologist. I study this as well, but I'm usually more interested in what's happening within the lake. And virtually everything living in the lake is leaving some sort of record in this history book. And so much of this is tiny fossils of organisms that lived. It's also geochemistry. It's a whole lot of other things. But a lot of what I'm going to be talking about is tiny fossils. The lake is full of microscopic organisms. Almost all of them leave a record in the sediments. The process is fairly straightforward. If you can reconstruct what lived in the lake, you can reconstruct how the environment of the lake has changed, because different organisms live in different types of lakes. Some live only in shallow water, some live only in deep water, some live only in salty water, some in fresh water, some live in acid water, some live in alkaline water, some like high phosphorus, like high fertilizers, some like only very clear water, some like cold water, warm water. And a large part of what my lab does is cataloging where these thousands of organisms live. And so we can actually do this in a highly quantified way. So the lake history book going back 12,000 years is full of information of what lived in the lake, what contaminants came from lake. We can reconstruct the terrestrial history from pollen grains. So we actually have a pretty complete history book of what happened in the lake. Now, as I said, we can go back 12,000 years. And we have gone back for Lake Opinacon, one of your lakes, we've gone back 12,000 years. We've gone back right to the Ice Age and studied it. However, the main focus here is on over the last 250 years or so. We call that the Anthropocene. It's when humans were major, European, certainly there were indigenous peoples in all these waterways, but their impact was minimal in these lakes. We cannot not see, right now, we do not see any impact of the indigenous peoples that were here. We do see when the settlers came, that's for sure. And to do that, for lakes in the Rideau Lakes, really we need about a core that's about 50 centimeters deep. Go down about 50 centimeters, about half a meter, you've got about 200-300 years of lake history for most of your Rideau lakes. How do we get that? We want to do that very carefully. Those sediments, that mud is very wet, very easily disturbed. If you mix up the sediment, you mix up the history book. We have these cores that were developed by John Glue. This is actually on Lake Opinacon, right in your series. That's my colleague Brian Cumming taking gravity core. These are the cores we use to take the surface sediments. They don't disturb the surface sediments. It's a fairly simple mechanism. It goes down on this rope here, you see, and it goes down. I use the analogy of surface sediments have about the consistency of a milkshake. That's how loose they are. But if you have a straw and you put your thumb on top of it, if you put your straw slowly into your milkshake, you put your thumb on top of the straw, you pull it out, you can take a core of your milkshake. That's more or less what we're doing. It goes down with this plunger here is in the open position. We slowly lower it into the very wet sediment. Then we send a lead messenger, sort of this messenger down this line, and it's like putting your thumb, it closes the mechanism, and up comes, we lift up the sediment core. And as you can see, even though it's very, very wet sediment, I think you can even see on this diagram how clear the lines are. It's definitely not been disturbed. Once we have it, then we have to section it. We want to go back in time. That would be 2021. We want to go back in history. There's the history book. The way we do that is we use close interval sectioning. Here we're sectioning at a 1/4 centimeter at a time. We're pushing this, it's very wet, so we have to be careful how we section it. And we're pushing it up from bottom. Here is where all the fossils, all the chemistry, all the history, it's a history book. I think of this as a time machine, and these like pages in a book that we're going to read going back in time. So we section it at a 1/4 centimeter. That's usually 2 or 3 years of lake history. Now, how do I know it's 2 or 3 years of lake history? Well, the first thing you got to do is you have to date the sediments. You have to find out, is this 12,000 years ago? Is this 5,000 years ago? Is this 50 years ago? How do we do that? Well, once you get into the thousands, we used radiocarbon or C14 dating. It's the same thing the archaeologists use. That goes back thousands of years. We take the sediment, we take usually seeds that we find in the sediment, we send them to Ottawa, and they do what's called a radiocarbon date. And they say, this sediment is 4,200 years old, and this sediment is 10,500 years old. That's for the long term. But radiocarbon doesn't work for the last 150 or 200 years. For the last 150, we need a different isotope, and we use lead-210. I won't get into any details, but lead-210 is a naturally occurring isotope, but it disintegrates every 20, its half-life is 22 years. So every 22 years, there's half the amount left. So it's a roundabout way saying that if I measure how much of this radioactive isotope is in the sediment, I can actually, instead of just putting, this is 1 centimeter, this is 50 centimeter, I can put dates on the sediment core. So this is about 1994, this is about 1981, this is about 1936, this is about 1850, and so on and so forth. So to put a Canadian context here, that's about the time of Confederation down here. And I love this one. This is the last time the Toronto Maple Leafs won the Stanley Cup. Yeah, you can see quite a bit of sediment has accumulated since then. But we can also use it as when cottagers arrived. We can use it when the Rideau Canal was built, a major factor in your part of the world. So we can actually put a time. It's not just random. We know this is about the 1980s. This is about the 1970s. Okay, so right now we have a history book. We have dates, but it's just mud, right? But in that mud is remarkable what's in there. And we look at all sorts of things. But some of the things, I'm only going to show you some of the things we find in sediments, and I'm only going to talk about 2 of them in this because of time. we can actually look. As you know, uh, people go to your lake and they measure how much chlorophyll A is in the water. That's to see how much algae is in the water. You don't want too many. I mean, you want algae, but you don't want algal blooms. You certainly don't want blue-green algal blooms or cyanobacterial blooms. That's something you don't want. Well, the in the chemistry of the sediments, there's ways of measuring how much chlorophyll A was there, how much total algal biomass. So I'll be using that today. There's also sometimes we can go and look exactly how much of different types of blue-green algae. I won't talk about that today, but we have the capability of doing that. The main thing we're going to be looking at is chlorophyll A and diatoms. What are diatoms? I'll talk more about them in a minute. But diatoms are the most common algae you typically find in your lakes. They're microscopic. You put like 50, you put hundreds on the head of a pin, but they're quite remarkable. There's thousands of species of diatoms living in these lakes. It's quite remarkable. And they're very special because their cell walls are made of glass, so they're very well preserved. I'll show you some pictures of them in a minute, but this is a major type of indicator we use because different diatoms live in very different types of environments. We can also look at benthic insect fossils and other types of invertebrate. These are water fleas. We do all these indicators in my lab. But today I'm going to talk mainly about algal blooms. So I'm going to be talking about chlorophyll A and diatoms. And so let's just say a bit more about diatoms because they're the key indicators. These are highly magnified pictures of diatoms. These are like high resolution microscopes. And this, if you sip some water in Lake Apinnagon or Indian Lake, you're drinking hundreds of these at a time, so that you're not going to choke on them. They're tiny. These are highly magnified, but I think they look quite remarkable. There's thousands of species, and they all live in different environments, and they're characterized by these glass cell walls, which is very important. So, and different species have very different structures. So, for example, this one over on the right bottom here, Cyclotella. This is one of the Cyclotellas. It's found only in very cool. It's found in cold, but very, very clean water. It can't take any type of pollution at all. Meanwhile, this Olacosyra here can tolerate a fair amount of pollution. Things like this Cymbella is only found on the macrophyte, very shallow water. It indicates very shallow water conditions. This one lives on the sediments in very shallow water. So this is what our lab does. We have this very quantitative. I'm doing this in a very sort of casual way here, but we can actually do this in a very quantitative way. And we're dealing with hundreds of these species. These are all not swimming around, but they're floating around in Lake Apinnagon, Indian Lake, all your lakes, and you don't really see them, but they're all there. Okay, so the overall approach is fairly straightforward. We go to something like Lake Apinnagon or Indian Lake, we take the sediment core, we section it, we date it, we look at all the fossils. This is a very long step. This takes a year for a core or something, so it's a very long step, and we analyze the data. Well, what can you do with paleolimnology? We do many, many, many things in our lab, but I'm going to look specifically at algal blooms, something that's in the topic, very much in the topic in the Rideau Lakes and elsewhere. Why are we getting algal blooms? Now, I point out eutrophication because eutrophication, as many of you know, is the over-fertilization of lakes. This is like coming from like fertilizer coming from agriculture or leaking septic tanks or sewage in some way. Algae need nutrients, just like a farmer fertilizes his or her field. The plants need fertilizer. They need nitrogen. They need phosphorus. So do the plants in a lake. And of course, if some of that nitrogen and fertilizer goes from the land, whether it's from your septic tank or from your agricultural field or from your golf courses, if it goes into the lake, it's no longer fertilizing the crops or the grass in the golf course. It's fertilizing the lake, and it's increasing the likelihood of algal blooms. Well, we got a pretty good handle on eutrophication and phosphorus and nitrogen, especially phosphorus. In the 1970s, new legislation came in, no longer having phosphorus in detergent. You had to have better septic tanks. And we saw real progress on algal blooms, real progress. What's been happening over the last 10, 20 years? Well, the nutrients are still low, but we're starting to see a reappearance of many algal blooms. And this is what I want to talk about. And so I'm going to use these approaches to do two things. What I say, targets, looking at what were systems like before we were here, before settlers were here, and what is the trajectory of changing? Can we get any clues from history as to what's going on? So we don't want cultural eutrophication. We just talked a bit about it. You add too many nutrients, or if you have too many algal blooms, you get algal growth, some of which are toxic health issues. You get taste and odor problems. It decreases property values. You get shoreline fouling, decreasing property values. You can get, once these algae die, they sink to the deep water. It uses up oxygen. It kills, it goes at no oxygen. You get fish kills, undesirable species, property values fall. And it's really, the real problems is not just algal, but a specific type of algae that people really worry about, so-called blue-green algae or cyanobacteria, that are especially in the news today. Well, we thought we had a pretty good handle on algal bloom, cyanobacteria, and we had progress. We definitely had progress in the post-1970s in dealing with this problem. Well, what's been happening? We've been seeing, this is a paper that studied some time ago, and it's going to be soon updated. But what we're seeing is cyanobacterial blooms are increasing in Ontario lakes, even though nutrients are going down. And in fact, most of the, one quarter of these are what we call oligotrophic lakes, nutrient poor lakes. This was not expected. Something has fundamentally changed in our lakes, especially since the 1970s. And what is that? Well, there's many possible factors, but and some of this data is preliminary. But we are finding, I work on climate change, and I work on many environmental problems, contaminants. Also, every problem I work with is worse with a warming planet. I think we really have serious problems in our system. So I'll just give you some background with this. And before I get into climate change as a factor for algal blooms, I want to start off. I gave a keynote to a scientific conference, and I realized I got to make this clear at the start. Without question, there's nothing that was wrong with what we knew in the 1970s. Without question, you need a certain amount of phosphorus and nitrogen and other nutrients to generate algal biomes. If you don't have phosphorus and the fertilizers, you're not going to get an algal bloom. But, and here's the important, people are now recording increased algal blooms even in lakes with steady or declining nutrient levels. And this includes some of the Rideau lakes. So we asked the question, can climate change be affecting algal blooms, even without increases in nutrients? And what I refer to here is the longer summer. With less ice cover, and this is documentable, we've been decreasing ice cover. I'm sure many of you know, if any of you go ice fishing, you know the time that you can ice fish is a lot less. If any of you are all year cottagers, you know that the amount of ice has been decreasing. This is very easily documented. It's one of the more visual signs of global warming. If you have less ice, you're making your summer longer. And algae are photosynthetic. They don't do much under a big ice cover, but they do a lot once the ice is gone. So the first thing is you're getting a longer summer, having more time for these algal blooms to develop. And lakes are becoming more and more thermally stratified. In fact, we have lakes that are shifting from before would be totally mixed in summer, almost the entire summer. Take Lake Opinacon. When I came to Queens, it was called what's called a polymictic lake. It was always mixing. It's only maximum depths about 9 meters deep. Wind would come. It wasn't deep enough to layer into layers. Lake Opinacon now is no longer polymictic. It's thermally stratified in summer. Just that small warming we've seen and the longer summer, Lake Opinacon is now a very, very different lake than it was 20 years ago, than it was 10 years ago. And what is thermal stratification? I think most of you know this, but I'll just say it briefly. Just like different liquids have different densities. And if you're making salad dressing, I guess two things you put in is vinegar and oil. And you have to mix it a lot. That's wind. Vinegar is denser, oil floats on top of the vinegar. If you don't mix it, you'll have an oil layer and a vinegar layer if using those. I mean, I could say scotch and water, but I don't want you to get into my habits here. And you have to mix it a lot. Think of that as wind. That's what wind is required to mix it. If you let the surface of a lake warm up, warmer temperature is less dense. It's like the oil. Colder temperature is like the vinegar, more dense. And so what happens as the surface warms up, it makes a distinct layer. And if you go out, if you're a swimmer and you're out treading water, sometimes in the middle of a lake, sometimes you're nice and warm and some of your toes are really cold. It gets cold like at your knees very quickly. that means your body, the length of your body has penetrated into the denser cold water. So using Lake Appinicon, lake I assume you're fairly familiar with, it's shallow. 20 years ago, that would not happen in the lake. It was just mixing all the time. Now with the longer summer, it's split into these three layers called the epilimnion up on top, the metalimnion is where it changes, and the hypolimnion where it's deep in the water. That not so places like Lake Appinicon gone from permanently mixing to stratified. Places like Indian Lake always stratified because it's deep. It's about over 20 meters deep, but now it's stratified longer and more strongly. So we've changed the thermal properties of the lake, and this is just perfect territory for algal blooms. I'll start with a quick example outside of the Rita Lakes where we started developing. I'm going to take you Lake of the Woods. Now, very big lake. Lake of the Woods is almost a Great Lake. It touches on two provinces, Ontario, Manitoba, and also into Minnesota. And you can see it from space. It's a big lake. It's a lake that we thought was a great success story with phosphorus. Back in the 1960s, this is the inflow of phosphorus, a major nutrient that drives algal blooms. And we, with the legislation in the 1970s, look at how phosphorus has greatly declined. In many ways, in the back in the 19s to the 1970s, it was like a success story. And yet, over the last few years, since, the last 10, 15 years, what's happening in Lake of the Woods? We're starting to get algal blooms and blue-green algal blooms. Well, we're getting complaints of blue-green algal blooms. And so we always worry when you just get complaints because maybe people didn't complain about it 30 years ago. They just thought it was natural. And now they're getting, well, there's a perception in Lake of the Woods that blue-green algal blooms, these are the ones you really want to avoid, are increasing. And we've got phosphorus going down. What's happening? This is not what we said would happen, or not what we expected to happen. We said we should have declining, and now they're creeping up. What's that? Well, what has been happening is we've had climate warming, and the lakes have fundamentally changed in structure. And I'm going to show you, the diagrams I show you are impossible to read because there's so many species. I've tried to really simplify them, but I'm going to take you to one bay in Lake of the Woods. It's such a large lake as bay. And these are just two of the major fossils we look at. This is Olacosyra down here, and this is Cyclotella. Olacosyra likes mixing. It's what used to be in Lake Appinicon when it was polymictic. Cyclotella likes the warm surface area. It doesn't like ice. So if you like, this is the cold water species. This is the one that's indicating better thermal stratification. You can see something this is the fossil record. That's the surface of the core. That's down about 200-300 years. That's the date 1980. I think you can clearly see there was a fundamental change in fossils in the early 1980s that continues to the present day. And we can count, we actually have temperature records for this area. In green is the annual temperature. In red is this is the green is actual measurements of temperature. The red is actually the fossil Cyclotella. Look how nicely it matches the known temperature. The inverse here is this is the cold water species. You see how it's totally opposite to the temperature. We also have ice out. Remember I said ice out's the clearer factor? Look at this. That's the ice out day, so less and less ice. With less and less ice in the longer summer, up comes this fossil. With less and less ice, down goes the one that likes ice. So we actually have a way of monitoring how the actual structure of the lake has changed. So the next question, so we now know these parts of the Lake of the Woods has changed dramatically. Have algal blooms increased in recent time, even though nutrients have not? Remember I said we can measure the amount of chlorophyll and its products in the sediments. Sure enough, if lakes are above about 10 parts per billion phosphorus, this is a proxy for algal blooms. Here's one part of Lake of the Woods. You can see starting in the 1970s, especially post-1980s, especially when climate warming really started accelerating, post-1970s, post-1980s, in this part of Ontario, up goes the algal blooms. If you go to just a slightly less phosphorus, it's less clear, but still you see an increase. So you still need the phosphorus. Phosphorus is key, but it seems you're going to need less phosphorus now to get an algal bloom. If you're less than 10, still pretty well a straight line, just a slight increase. So what is clear here is that if nutrients are still very low, you do not get big changes. But even when nutrients are still pretty, reasonably low, we are now seeing algal blooms. And especially we're seeing blue-green algal bloom, because blue-green algal blooms love the longer summer. They love the hotter temperature. They love the thermal stratification. They're completely adapted to living in these stratified environments. When you're going basically from here, so this is what Lake of the Woods would have been like in the 1960s, 1970s, or Lake Oppinicon was up until about 10 years ago. And now we're going to these stratified systems. This is what Lake Oppinicon is now, if you'd like to use an example. This is what the bays I showed you. Could exasperate the likelihood of bloom, even without further increases in nutrients. Okay, so I haven't talked about Rideau Lakes, and I'm going to start with just two quick Rideau Lake examples. I'm going to talk about Lake Oppinicon and Indian Lake, the two we've been working on so far. I want to say that everything I'm showing you is preliminary data. I'm showing you what is coming off the microscopes now, so we can't, these are not finalized data. This is part of CAP's master's thesis, but I'm going to take you to the lakes and since I'm talking about the Chaffee's areas lakes, the two that I'm going to talk about is Lake Oppinicon and Indian Lake. Two very, very different, two lakes side by side, very, very different lakes. Lake Oppinicon is shallow and full of macrophytes, full of aquatic plants, macrophytes. A lot of people don't like those aquatic plants, but they're pretty important for the lake. Indian Lake, a much deeper lake, has had a very different history than Lake Oppinicon. Let's start off with Lake Oppinicon. It's a long lake, narrow. Here's the key. It's a shallow lake. It used to be polymictic. It's so shallow, most of it's only 2 meters deep or so. It used to be polymictic. Up until about 10 years ago, it was mixing. It's now warm enough that in summer, it's stratified. This is a fundamental change in the lake. It's meso-oligotrophic. The nutrients are quite low, but they're in the danger area. And what's important, it's macrophyte dominated. A lot of people, by macrophytes, we mean those aquatic plants that some people call seaweeds. They're not seaweeds, but they're the plants near the shore that a lot of people don't like, but they're pretty important. And then afterwards, I'm going to talk about Indian Lake, very different lake, 26 meters deep. The main, again, I'm going to say I'm mainly talking about diatoms. I talked about these earlier. We've got lots of indicators, but because of time, I'm going to talk about diatoms. Okay, what we did, I'm going to look at Indian, and I'm going to start off with Lake Oppinicon. Back 25 years ago, I cored Lake Oppinicon and Indian Lake and other lakes in the Rideau system and published on them. I did this with different students. This is early in my career, 25 years ago. And what we're going to do now, what Cap is doing for his master's, he's using the same, well, we have new methods, but the same types of methods. He's gone back 25 years later and cored the same lakes. So Cap, in his new master's, that's funded partly by the Rideau Lakes Association, he's going to see what's happened in the last 25. He's going to really focus on what's happened in the last 25 years, the period of accelerated climate warming. So first I'm going to show you cores I took 25 years ago. These figures are, I agree, impossible to read. I'm not going to ask you to look at the detail. I'm going to look at the general pattern that we see. Okay, so this is the core we collected in 1995. So can't believe it, but it's 26 years ago. I remember taking the core. So this is Lake Oppinicon. I should have said Lake Oppinicon there, but trust me, this is Lake Oppinicon. We published it we published it over 2 decades ago. This is the history. I know this is impossible to read. I just want to show you a few things. This is the history book. That would have been 1995, the surface of the core. And this goes back to the early 1800s. We actually went back 12,000 years, but I'm just showing you this. What is remarkable, and these are the different fossils that we see. The main ones, these are just the dominants. The main point is that we see virtually very little change in the fossils. We had a massive thing happen in Lake Oppinicon in the 1830s, 1820s leading 1830s. The construction of the Rideau Canal. We see big changes in the other Rideau lakes, the deep lakes in the Rideau Canal. Lake Oppinicon was dramatically changed with the building of the Rideau Canal. It more than doubled in size. They flooded the local land. It completely changed the lake. It was a much shallower, much smaller lake in the early 1800s before they dammed it up because they needed it big enough to get the ships through. We see Very little change in the fossils in Lake Opinion. We were very surprised, but it made sense. In fact, this is a bit what we call a complacent profile. There's a little change in plankton. The lake got deeper, and sure enough, we got organisms that like deep water. But we saw very little change in Lake Opinican. This really surprised us because there were tremendous changes when they built the Rideau Canal. They flooded areas and they cut down remarkable numbers of trees. Erosion, they needed it for the building. This is about this is the Kingston Mills, not far away. Look at that. There's virtually no trees left. Even if you look at some of the photographs, this is about 1832 on Upper Rideau Lake. Virtually all the trees are gone. It was denuded. And yet Lake Opinican had remarkable resilience. And the reason it did, and we understand that pretty well now, is we can tell that from the fossil record. Lake Opinican always had lots of aquatic plants, lots of aquatic macrophytes. And we now know that they're incredible stabilizers to a lake ecosystem. I know a lot of people don't like them. They don't like swimming through them. They don't like boating through them. They don't like their fishing like they do. But they actually suppress algal growth because they're competing with algae for the nutrients. They reduce nutrient availabilities. They also reduce substances that deter other organisms and their habitat for fish and stuff. So Lake Opinican was remarkably stable even with those disturbances because it was full of these aquatic macrophytes. Okay, that was 25 years ago. What's about climate warming and the other stressors hitting Lake Opinican? So what we did in 2019, Cap went and we took 25 years later another sediment core. And there's Cap sectioning his core. What's happened in the last 25 years? Well, if anything, nutrients are about the same, but if anything, they've declined. It's good news. You've done a very good job if you're on Lake Opinican to declining nutrients. That's true of many of not all the Rideau lakes, but many of them, it's about steady or declining nutrients. Yet algal blooms are being recorded. Warming definitely has been accelerated. And we're dealing with a very strange lake in Lake Opinican that used to be polymictic and now is stratified in summer, stratifies in the big warning sign. Do we capture this in the fossil record? So Cap has gone and taken new sediment cores and he's trying to combine it to what we knew when we took cores 25 years ago, but we're adding the top 25 years of material. Well, again, these cores this is preliminary data. Please don't take it. But we now also have new techniques like the chlorophyll A. Is there any warning signs? Well, first of all, that's before the Rideau Canal was in. That's prior to the Rideau Canal, more or less pristine conditions. And like I said, it was a shallow system. We know it was shallow, but the prospector said it was shallow. The engineers said it was shallow. But these are all shallow water species. It was all except Lancashire. It was a shallow water lake, and it was full of macrophytes. Then they built the Rideau Canal. This is the post-Rideau Canal. And we do see some changes. We saw that it flooded the area. You see these are areas. We saw some changes, but again, remarkable stability in this lake, given all the disturbances. There was agriculture, everything, probably because of those macrophytes. It survived the Rideau Canal. It survived all sorts of things. Look at what's happening in the last 25 years. Look at the last 25 years, even though I'm sure these names don't mean anything to you, we are seeing changes. The lake is fundamentally starting to change. These are all consistent with climate warming. They're all consistent with increasing less ice cover and increasing stratification. Here's a summary diagram. Let's just look at, also to worry about, look at what's happening chlorophyll A, possible start of the worrying sign. This is just the same data. I put it in. I know those figures are impossible to read. I mean, I find them hard to read and I do this every day, but here, these are the shallow water diatoms. These are the open water diatoms. These are the ones that don't like stratification. And this is the total amount of algae present. So in the past, before the Rideau Canal, the lake was quite productive. And that makes sense. The lake was much shallower. It was light all the way through. There was macrophytes everywhere. With the Rideau Canal, it actually got flushed out a bit. And so the chlorophyll A actually went down. Not surprisingly, the plankton change. But what's really critical is what's happened since warming. You see the chlorophyll A starting to peep up. The one that likes mixing declines. These are the algae that are showing us that we're thermally stratified, and the benthos is decreasing. I would say these are warning signs that Lake Apinnaconn is shifting and you have to be careful. What we're seeing is this happening in Lake Apinnaconn. Lake Apinnaconn back 20 years ago was like this. Now it's going like this. This is, we've seen this before in other lakes, and it's the early signs of warning. So we went from Lake Apinnaconn, a shallow lake, macrophyte dominant. It shifted from polymixis to thermal stratified, longer summer. That's a strange lake because we don't usually work on such shallow lakes. What about Indian Lake? Right next door. So anyways, sharing water to some extent. Very different lake, 26 meters deep, not macrophyte dominant, doesn't have that resilience. It was always thermally stratified, but now more strongly, and now less ice cap. Let's take a look at Lake, the Indian Lake. This is the core collected. This was a core collected 21 years ago and published. And this is going to be a different scenario from Lake Apinnaconn. Remember, this is the deep lake. This doesn't have the resilience of the macrophytes. So in blue, I did so this is the building of the Rideau Canal, this line. Pre-Rideau Canal, it was a clean lake, nothing here much to worry about. It was like one of those pristine deep water lakes. Came in with the Rideau Canal. This one didn't have the macrophytes. Things to worry about: Fragillaria crotonensis, Alacasaura ambigua, other species that are warning signals that the lake became significantly more productive, probably a higher likelihood of algal blooms. What's happened since that time? So this is still the old core. This is the 20-year-old core. Then in the 1970s, as you know, we brought in phosphorus regulation. Everyone got better septic tanks. They took phosphorus out of detergents. And you see some very good news happening. It's sort of a returning to clean water conditions post 1970s. You see these are diatoms that like clean water. You saw Fragillaria crotonensis decreasing. So this was sort of a good news story when we published this paper 20 years ago. What's happened in the last 20 years? Again, very preliminary data, but CAP is finding, again, some signs of worry. With the warming that's happened in recent, look at that, Fragillaria crotonensis. That's the one I said we had something to worry about back there. Look, it's definitely increasing. This is typical of a longer stratification. Also, look at what's happening to chlorophyll A. It's not horrendously high, but again, worrisome. Other species are changing, totally consistent with the longer summer. So this is preliminary data, but since I was talking about the Chaffee's areas, I thought I'd talk about the two lakes we had. There are concerns. With decreased ice and snow cover, we have a longer summer, allowing more overall algal production. In addition, with the warmer summers, we're having this thermal stratification is more intense, it lasts longer. For some lakes, like Lake Apinnaconn, it's gone from not stratified to being stratified. Wind speeds are also declining, by the way, which is also helping the cyanobacteria. It's a perfect storm in many ways. And these conditions allow cyanobacteria to thrive. And we are now seeing blooms, and especially cyanobacteria blooms, occurring in lakes with increasingly lower phosphorus concentration. Management implications: managers, people who take care of lakes, you're probably going to have to set even lower phosphorus guidelines, and that's not good news because it's been very hard to get to these phosphorus guidelines. As climate warming continues, and it's going to continue because we've been very bad about dealing with this terrible problem, this is just going to be one more of the things we have to worry about. So lessons from the past, unintended consequences. I mean, you think you solve a problem, then you add another problem, and it sort of resurrects a problem that you thought we kind of had under control. And the one thing I find in my field, we tend to be overly optimistic. Things are generally worse and more complicated than we thought they were. And then finally, my last slide, history is important. We deal with history, and I think mostly think of monitoring as days, maybe going back in years. And if you think of paleoecology, you think of thousands of years or centuries. But these new approaches, they're happening in between. And I think a lot of the important answers lie in this area. So thank you very much. And I'm happy to take questions if there are any, I think I'm pretty well on time, I think. Thank you.